Started, yeah.
Okay, good, I'm starting. I have a teleprompter. Good evening and welcome to the 5th webinar of the Indian Pollinator Initiative. My name is Axel Brockmann and I'm one of the 5 organizers of this seminar series.
Started, yeah.
Okay, good, I'm starting. It's my pleasure to introduce today's speaker, Professor Deborah Smith. Good evening and welcome to the 5th webinar of the Indian Pollinator Initiative. Oh, wait a minute, stop. My name is Axel Brockmann and I'm one of the 5 organizers. I have to watch YouTube and Zoom to see the questions on the YouTube. And the YouTube started without, and I had to stop it. Okay, sorry about that. So, okay, it's my pleasure to introduce Professor Deborah Smith from the University of Kansas. Deborah is one of the most knowledgeable researchers on honeybee diversity. She did her PhD at Cornell University under the supervision of George Eichward, who is a leading expert on Halactidae, that means sweat bees. And then afterwards, she joined the group of researchers around Charles Michener at the University of Kansas. Charles Michener, everyone knows it, is a leading figure of bee systematics. So she was at both big pillars of bee systematics, Cornell and Kansas. Another fact I would like to mention is that in 1991, and that must be early in her career, Deborah was the editor of a book with the title Diversity in the Genus Apis. In my opinion, this is the starting point of all the recent comparative studies on Asian honeybees. And I have to admit that it also got me started working on Asian honeybees when I was a student in Germany. Of course, the organizers of the seminar series hope that this seminar series will do the same for the students and younger colleagues in the audience. Before starting, let me remind you about the procedure. Participants on live size will be able to raise their hands and ask then their questions directly. Watchers on YouTube can write their questions in the chat box, and their questions will be read by me. We will have a short question and answer session in the middle of the talk and a longer one at the end. Without further ado, Deborah, it's your turn to talk about phylogeny of Apis species and the honeybees of India. Welcome.
Thank you. Well, let me project this. Here we go. So, as I say, today I'm going to be trying to summarize several decades of work on Apis species, particularly in Asia. And I want to tie it back to India because there are a lot of interesting and unique features, as it turns out, about the honeybees of India. So if you are working with honeybees, you probably know very well already that there are 3 main groups within honeybees: the dwarf honeybees, giant honeybees, and cavity nesting bees. And all of these are Asian. Oops, these are Asian except for Apis mellifera. So over the years, I've been doing genetic studies of honeybees using, at first, data from mitochondrial DNA, which included restriction site polymorphisms and sequence data, and more recently, genomic data, both sequence and in particular, single nucleotide polymorphisms, or SNPs. And all of this was with the goal to understand the diversity and evolution of honeybees. So in the first part, I'm going to focus on dwarf bees and giant bees. So using primarily older data sets on sequence data from cytochrome oxidase 1 and 2. Cytochrome oxidase 1 is sometimes called the barcoding gene because it has proved so useful for identifying differences among species. And we'll look at the dwarf honeybees and giant honeybees, touch on the special implications for Indian dwarf and giant bees, and then stop for questions. And then after questions, go on to the cavity nesters, Apis cerana, and the other Asian cavity nesting bees.
Deborah?
Yes.
So we cannot see your slides.
Oh, you can't? Oh. Let me try.
I was not yet sure whether it was the intro.
Okay, let me try that sharing again. Maybe we lost the share. Okay. So okay. There's the share. Okay, it should be sharing. And do you see it now?
I can see it.
Okay, I'm going to put it onto slideshow. And do you still see?
Oh, no, now it's gone.
Oh, for goodness sakes. Well,
wait a minute, let me check. Emma?
Yeah, that's interesting. Okay.
Can you try to share it again?
Yeah, do it again.
Now something is happening. Yeah, now we can see it.
Right, but if I put it on like this.
Yeah.
You can't. So I think you can, yes. So it's gone away.
Okay.
So if you can stop the slideshow mode.
Sure. Yeah, that's too bad.
Then I think it works.
The other day. Okay, so I will just scoot through these introductory slides. We looked at the subgroups within Apis, the dwarfs, the giant, and the cavity nesting bees.
Deborah, we can't see it again.
Again, it keeps disconnecting itself. It did not do this when we practiced it. Let's try. Goodness.
Happening now.
Happening.
It's fine. Yes.
And so
I see it.
Then maybe just kind of increase the picture on your.
Yeah. There we go.
Yeah.
Okay, now let's see if I can do this without losing it. So, okay, so I said our goal, we're using genetic work to look at diversity and evolution of the honeybees. And I mentioned that we will be looking at the dwarf and giant bees first. So starting with, is it still showing? Still visible, okay. Starting with Apis, the dwarf honeybees, we have two recognized species, Apis florea this is an example from Thailand, and Apis andreniformis, an example from Borneo. And this map shows the distribution of the two species with blue dots for andreniformis, orange for florea. So you can see that florea has a much broader range than the more eastern Apis andreniformis. Apis florea is also one of the more invasive honeybee species. All of these localities indicated by stars are extensions from the natural range of Apis florea. So it's been moving westward into Africa, and it's been probably transported by people to some populations, even as far east as in Taiwan. So earlier in a paper by Randall Hepburn and other authors, they made the hypothesis that the speciation event that led to florea and the more eastern andreniformis took place somewhere in this region with probably the Arakan mountain range, serving as a barrier that led to divergence between the populations to the east and west. And they could have become a barrier to movement of these bees during historical periods when the climate was unfavorable for open nesting tropical bees. Oh, I have to do this by expanding. I've lost my, well, we'll just do it this way, go there. So we'd use cytochrome oxidase 1 and 2 genes to examine diversity within and among these dwarf bees. We found, of course, we used outgroups of stingless bees and bumblebees, and we got nice support, thank goodness, for Apis andreniformis as a species. Samples from Thailand, Malaysia, Borneo, Palawan, and very good support, 99% bootstrap support for Apis florea as a species. That's good. But you'll notice within Apis florea, we found two highly supported, also 99% support for two groups within florea, a Western lineage that included our samples from India and Iran, also Saudi Arabia and the introduced Israeli and Jordanian populations, and then an Eastern group that included Thailand, Cambodia, and actually I left off Laos. So the boundary between the East and West is also, we have samples from India and points west. We have samples from Thailand and points to the east, but we don't have any genetic samples from this critical region up here between where the Arakan Mountains is a probable barrier. So we suspect that this is probably a space where, for a second time in history, Eastern and Western populations of dwarf honeybees became isolated from one another for some length of time. They differentiated, at least in their mitochondrial genomes, and it's a very interesting question to ask whether or not these are now two cryptic species, two separate, physically similar species. So we'll come back to those, but next to look at the giant honeybees, I say 1 to 5 species in this group, and that's because there's a lot of controversy about these bees. So anyone who's living and working in India is probably very familiar with these bees. In fact, this picture is from some bees in the Bangalore area. Yeah. So this is work that was done by an undergraduate honors thesis student of mine, looking at the distribution and relatedness among giant honeybees. And her basic question was, in this range of giant honeybees, how many species are there? So there are two extreme hypotheses. At one extreme, there are people who feel that all of the giant honeybees are a single species, Dosata, and that all of the divergent populations within them, such as Laboriosa, the rock bee, the Breviligula bees in the Philippines, Binghamai bees in Sulawesi, they're all subspecies of a single species. Earlier authors have proposed each of these as species. And I would say that only the most diehard single species, non-splitters, no longer accept Laboriosa as a full species. It's very well documented now. But at the other extreme is the proposition that there are as many as 5 species. So essentially each of these subspecies raised to species status. But here's the surprise. An unnamed species at the moment, what appears to be an unnamed species in the south of India. So where did we get that notion? And it's probably no surprise that this is my hypothesis. So our student who did this job included samples of Laboriosa from Nepal and 28 samples of Dorsata in the broad sense from the Philippines and Sulawesi, and the broader range from Pakistan and India, Nepal, all the way out to Indonesia. She used dwarf honeybees and mellifera as outgroups, sequenced 50 sequences of about 1,500 or more base pairs, and did a maximum likelihood analysis. So this just shows the locality of our samples. I'm already just assuming Laboriosa is a good separate species, but you'll see that is the case. So samples ranging from Pakistan over to the Philippines and in eastern and Indonesia. So here is the tree that she came up with. So these are the outgroups, the dwarf honeybees. And here is Laboriosa, Dorsata in the broadest sense, and Apis mellifera. And the 2 points to notice about this are, first, that this fact that we have essentially 3 branches coming off a single node here, this means that it recognizes 3 groups, but makes no claims about how closely they're related to each other, which, since we've just got 2 giant bees and mellifera, it's basically saying mellifera is different, Laboriosa is a distinct species, and Dorsata, this large group, is another lineage. So, oops, gotta come here. Both the behavioral, morphological, and now genetic data showing that Laboriosa groups entirely outside of all of the other giant honeybees, it's pretty obvious Laboriosa is a distinct species. And there are other studies waiting to be published by other authors that are going to demonstrate this very nicely. So zeroing in on the Dorsata in the broad sense group, I've color-coded each of the distinct branches that were found. One with moderate support is the oceanic Philippine population. This includes bees from the islands of Mindanao and Luzon. Sulawesi, we had only a single representative of Apis binghamii, or Apis dorsata binghamii. And then this is the more narrow concept of Apis dorsata. But the big surprise is that the samples from the south of India clearly came out with very high support as a separate branch, as distinct as the ones from the Philippines or from Sulawesi or the broader Apis dorsata in the broad sense. Now, if you have very sharp eyes and you're really looking at this, trying to read it, you might say, wait a minute, you have bees from India over here too. And that's true. So here, these navy blue stars indicate samples from India, but they are from the Andaman Islands, from Assam, here and here. So extreme northeastern India. And also we have samples of Dorsata from Nepal, clearly distinct from Laboriosa. Pakistan falls in this population. And if you're really paying attention, trying to read these names, here's one from the Philippines. But again, it's sort of the exception that proves the rule. These are from what we call the oceanic Philippine Islands, islands never attached to the mainland of Asia. This is from the island of Palawan, which both geographically and biologically is closely associated with Borneo, and at one time was connected to Borneo. So part of this larger mainland type group. So this color coding shows the distribution of those groups. So we have Laboriosa, which extends, if I had samples, all the way over into Vietnam in the highlands. We have the oceanic Philippine group with distinct sequences. We have the Sulawesi bees, the broadly distributed, what I would call Dorsata in the narrow sense, indicated by blue, and then a distinct population in southern India. So just briefly, I want to show you this illustration. This doesn't really pertain much to the Indian story, obviously, but it does give us a little more information about the bees on the oceanic Philippine Islands and Sulawesi. This illustrates what the world would have looked like 21,000 years ago at the last glacial maximum, when sea levels were 120 or more meters lower than at present day. And that illustrates the outlines of what is today a drowned continent, Sundaland. This is a shelf extending southwards from East Asia. For most of the last 60 million years or so, most of this land was above water. And there were periodic incursions of seawater into this area. If sea levels rose, more land is exposed during periods of glaciation, when much of the world's water is tied up in ice sheets. But although Borneo, Sumatra, Java, all of these islands were incorporated into the Sundaland landmass and thereby connected to the rest of Asia, these outer Philippine islands have never been connected to the mainland. They've always been isolated by deep water channels, the same for Sulawesi. And in fact, these are very young islands as well. So until about 10 to 12 million years ago, they weren't there as above water pieces of land. So they have been assembled over millions of years from various volcanic arcs and bits of land that have rifted off from other land masses. So it's perhaps not surprising that these bees have been much more isolated from the bees of Sundaland. They were colonized undoubtedly by bees from Sundaland. So if you look at the ones from Sulawesi and the Philippines, they're quite distinct from the bees the giant bees elsewhere. We're used, if you're living in India, you're used to these nice yellow and brownish bees. In the oceanic Philippine Islands and Sulawesi, they're distinctively black with white stripes. There are physical differences between the two island groups, but they have similar appearance and similar behaviors. For example, the bees in India and elsewhere in Asia are known for forming clusters of nests. And apparently, at least in the Philippines, the bees are not known to do that. So they have some behavioral differences, they have obvious physical differences, and they have genetic differences. So to sort of summarize this, we have mitochondrial data that certainly supports Andreniformis and Florea as species, but suggests that there's a possibility of a second, much more recent speciation event giving rise to two types of Florea. Within the giant honeybees, and I'm drawing this in a bit more detail than my students' cytochrome oxidase 1 tree, because we do have some SNPs data for giant bees, we have Laboriosa as a separate species from the whole Dorsata lineage, but within Dorsata, there's very good evidence that each of these should be recognized as a separate species. And in particular, the bees of India, the Dorsata from southern India, the Florea from India, are not associated apparently with the bees to the east. They're distinct from the bees of more eastern areas of Asia. So the next topic will be the cavity nesters. But just stopping there, what does it mean? So does this prove that there are 2 species within Florea, or that there's 5 species of giant honeybees? No. These data, we need more thorough genetic studies with larger samples for these, and in particular, behavior and morphological studies to really confirm whether each of these things we've discussed merits a species status. But the genetic data strongly support the hypotheses that there are multiple species currently lumped under Apis dorsata and possibly 2 species lumped under Florea. So in addition, the Indian populations appear to be distinct from those in Eastern Asia. So at that point, I can stop and ask if there's any questions.
So let me, okay, Sino has a question. Sino? Sino? Hi, Sino.
Hello, am I audible?
Hello.
Hello, Deborah. How are you? I hope you are doing well.
Yes, thank you.
Oh, okay. It's good to know that Apis dorsata of South India may be a new species. And it's also interesting to note that this oceanic Philippine islands have multiple branches, although they are very close to each other. My question here is, have you analyzed any species, any specimens from Sri Lanka, and how that appears, or what do you propose for that?
I have not had any samples of dwarf bees or giant bees from Sri Lanka. I do have some for cavity nesting bees, which I mention, but I would suspect that they would be similar to South India, which probably wouldn't surprise any biogeography person. But I don't have any samples to confirm that.
All right, thank you.
Okay, then there's a question by my colleague Sodamini. Sodamini?
Hello, yeah, very interesting data, genetics data on honeybees. And I was intrigued to I was intrigued to see the central in South Central area, how most unique species of honeybees that to be somewhere in Karnataka, Bangalore region maybe, but
yes,
yes.
Yes.
I wonder whether there were samples from the Western Ghats, Kerala, Goa, those sort of regions as well?
Yes, there were a few in there also from the Western Ghats.
Also have this unique position in the phylogeny, I wonder.
Well, the few I had from the Western Ghats, they were mostly due west of Bangalore, head west from Bangalore till you come to the mountains. But they were of that same southern type.
Oh, very interesting. Yeah. Thank you.
Okay, Ulasa.
Hi, Deborah. Have you done any molecular dating analysis?
Interesting, you should ask. on the next part that I'll mention, we've, when I talk about the cavity nesting bees, that is a dated phylogeny, and we only included dwarf and giant honeybees as outgroups, but that did give us a little information on the possible age of some of the groups.
Okay, that will be interesting. Thanks.
Okay, so Deborah, there are two questions on YouTube. From Saket Shotri. He was asking, where does Apis cerana indica lies in this tree? But I guess that you will talk. Yeah. And then, then there's a question from my colleague from GKVK, Professor Shakaband. But does Dosata in India are able to migrate long distances? Cause genetic mixing across kind of like the species? So,
well, yeah, that's a very interesting question, because of course they're known for that extensive migration, and this is why it's so fascinating that it seems there's another type. So one, as when I come to the very end of the talk, I will say, you know, one of the things we desperately need is systematic sampling across India to find out, like, what is the range of this genetic type, if it is a species, you know, where does it come into contact with the more northern, you know, so I have specimens from Nepal. I know they're different from the ones from South India. But that may even indicate that they have different migratory routes. So I know a lot of the work on migratory behavior of the Indian giant bees has been done up in the northeast of India. And that, pardon?
Go ahead. Then
I was going to say, that region is out, you know, as we'll see, that seems to be a region, at least if you look at the dwarf honeybees and the cavity nesters, that northeast of India is more closely associated with the east of Asia. And I think that's true for a lot of other organisms as well, not just honeybees. That's a biogeographically different region. So it could be that, you know, they have different migratory routes compared to the more southerly ones as well.
Yeah, so probably the southern Dosata will migrate from the west to east and not south to. So as you are saying, the migratory routes might not overlap, but one has.
We don't know until we look to find out.
Kind of like there's several other questions. You observed some difference in the nesting behavior and morphology of Apis florea and Apis. Have you observed differences in the nesting behavior and morphology of Andreniformis and florea?
Andreniformis and florea? Yeah, those morphology and nesting differences are well known. There's the color differences. The florea is a more bright orange and black stripes, whereas Andreniformis is more, as the fully colored adults are more dark. There's differences in the male, the drone hind legs that they use in mating or in holding on to females. The nests are attached to their substrate, to the branches differently, and there's differences in their dance. That's nothing that I've done. That's not my work, but I mean, those have been well studied. And so as those two species are well distinguished. Now, Eastern and Western florea, no, I don't, I mean, at this point, I don't know any morphology differences, but again, nobody's really looked either. So it would be interesting. And it may be that the differences are more in behavior, like mating times, or in the types of they seem to be very sensitive to what types of Varroa are hosted. There seems to be a close relationship, and that is often an interesting signal that there's something different about two populations of bees when you see they have different parasites.
Yeah, so there's a similar question regarding
I'm afraid I can't hear you.
You know?
Now I can hear, yeah.
So there was a similar question regarding Dosata, whether kind of these two Dosata types differ in morphology or behavior.
Again, that's something somebody working now in India needs to look at, because you know, I have not seen any reports of any physical difference. When you look at them, there's no obvious differences. Yeah, I assume it's going to take study of genitalia, male and female genitalia, but I'm not there. So someone who is there in India could take that on. Yeah.
Okay, let me. So there's Paul Williams on live size. Paul. Yes. Paul? We cannot hear you. Paul, you have to unmute your microphone. Okay, let's go. Paul?
Hi, Axel. Hi, Deborah. Really
Hi.
interesting. Can you hear me?
Yes, I can.
It's really interesting. Can you hear me?
Yes, I can.
Ah. Yeah, it says it's unmuted.
No, we can hear you, Paul.
I can hear you. Maybe you can't hear me, but.
Okay, let me kind of ask then a short question in between till Paul gets back.
Can you hear me now? Because there seems to be a very long delay.
We can hear you.
Okay, I think I'm going to send in an email later because there's a very, very long delay.
But you can ask the question so that we hear the answer.
Yeah.
Okay, he doesn't. So,
okay.
So let me ask you one question. Do you think that Apis laboriosa is ancestral in the group of the Dosata group?
No, I think that you know, there was one split that gave rise to the Laboriosa. That's an early split, yes, between Laboriosa and the other giant bees. But I don't know if you would say their ancestor. Some unknown extinct bee was the common ancestor to both of them.
Okay, I'm asking you because of Laboriosa, I mean the ocelli are more similar to all other honeybees, right? And then Dosata has these larger ocelli and can fly in the night. Laboriosa probably cannot do that.
Yeah, but I wouldn't call them, you know, you could say that they have retained that ancestral Apis character and the other lineage has not, but you know, they've diverged from a common ancestor. Yeah.
Thank you. I think, okay, Paul will write you an email and me, Paul. So I would like to hear your question. So, okay, good. Deborah, then I think you can go ahead.
Okay, here we go. Okay, so in this section, I'm going to focus on the cavity nesting bees, where I will start with a little older data from cytochrome oxidase 1 and 2, but mostly talk about the results of a genomic study. We're going to focus on the apis, so focus on the cavity nesting lineage within honeybees. And I just had to throw in a picture of Apis koshevnikovi. If you've not seen pictures of them alive, they are gorgeous bees, just beautiful. And so currently, within this group of the cavity nesting honeybees, the species that are recognized are mellifera, the western species, Serrana, the eastern hive honeybee, nigricincta on the island of Sulawesi, koshevnikovi, which is basically a Sundaland species. It occupies now remnants of Sundaland, so peninsular Malaysia, Sumatra, Java, and Borneo, and then Nuluensis, which is in the highlands of Borneo. Now, both the western and eastern mellifera and Serrana have very large ranges. They have multiple, very distinct mitochondrial DNA lineages, both allopatric and especially in the case of Serrana, some examples of sympatric populations with different mitochondrial lineages. And so we're going to focus on primarily the species within, or the populations within this species, Serrana. So here is a summary of older work. At the time we started this, nigricincta wasn't accepted as an independent species. By the time we finished, not because of any work that we did, nigricincta was then accepted as a full species. And these colors represent distinct mitochondrial lineages, not individual genotypes. So for instance, blue dots indicate what we call the mainland mitochondrial lineage. There's a lot of diversity of haplotypes within this group, but they're all clearly a distinct family of haplotypes compared to these other color-coded populations. So these mainland groups, we find all the way from Pakistan and India to Japan. There's a group that is the Sundaland, represents the Sundaland population, all the way out to some of these adjacent islands, and apparently sneaking, unless somebody carried the bees into Myanmar, that we suspect they're going up the east coast of the peninsula, getting in just to the bottom of Myanmar there. A Philippine group, and nigricincta on Sulawesi. And here is the group that person was asking about. There's a separate group there's two groups within India, and this has been known for many, many decades, if not centuries. The fact that there's a black version of cavity nesting bees, sometimes called the hill bees, and we found those to belong to the mainland mitochondrial group, and then a yellow bee, or the plains bee, and we found that in both southern India and Sri Lanka, a completely separate mitochondrial lineage. So within what is currently called Apis cerana, we've got a mainland lineage, a Sundaland lineage, Philippine and India yellow or plains bee lineage. And as long as as I'm showing distributions, I'll just point out here's Kashevnikovi with this nice Sundaland distribution. Its northern boundary is the boundary of seasonal tropical rainforest to the north and non-seasonal tropical rainforest to the south. And then Nuluensis, which we sadly did not include in our study, we weren't able to include it, occurs in the highlands of Borneo. So our main question is, are those mitochondrial lineages that we see within Serrana distinct species or not? So we used data from the nuclear genome, single nucleotide polymorphisms or SNPs. We generated those using a method called RADseq, restriction site-assisted DNA sequencing, and included dwarf bees and giant bees as outgroups, and then 86 samples of cavity nesting bees. And this is the point where I show you a tree, and I'm supposed to say, "Oh, I know you can't read this, but ... So I know you can't read that. So we'll use this pared down version of the tree. It basically shows that we recovered Micrapis, or the dwarf honeybees, the giant honeybees, and the cavity nesting honeybees, Florea and Andreniformis. Here we have Breviligula, Binghamii, Dorsata in the narrow sense, and the South Indian giant bee, Mellifera, and then the lineage of Asian cavity nesting species. So the dotted lines indicate species that we did not include in our study, in this particular study, but based on phylogenetic work by other authors, we're interpolating where those, approximately where they would fit on that tree. And this is a time-calibrated tree. So there's a time scale here going from the Oligocene to modern times. It's dated using a fossil specimen, Apis lithohermea, which is dated to 14 to 16 million years ago. And it is a giant honeybee. It falls into this, morphologically into this group of honeybees. So we use the date of that to sort of anchor how far back in time or forward in time the root of this tree would need to be to have a full-fledged giant honeybee at this time, when we know we also have to allow time for the evolution of the dwarf honeybees, the giant honeybees, and the basal members of the cavity nesters. So this is a bit older. This pushes the origin of the cavity nesting bees and honeybees in general, the extant honeybees, a little further back in time than some other studies that don't use fossil dating. And we could talk about why we come up with different dates if you don't include a fossil, one or more fossils as hard date points. So what I'll do is just walk through that tree, indicating what we think the set of events, what that tree implies. So at around 20 million years ago, a speciation event that gave rise to the ancestors of Mellifera and the ancestor of the Asian cavity nesting species as a group. And then about 15 million years ago, another speciation event that gave rise to the Sundaland, Kashevnikovi, and the other Asian cavity nesters, the ancestor of those bees. excuse me, which was probably in southern Asia or even partly in Sundaland. Next, a speciation event that stemmed from colonization of bees into the oceanic Philippines and Sulawesi, giving rise to Nigricincta and its sister group, the Philippine cavity nesters. The other lineage on the Asian mainland and Sundaland, oops, come back, gave rise, again, a speciation event to the yellow Indian form of cavity nester. And the mainland, the very last divergence was between mainland and Sundaland populations of Serrano or other cavity nesters. So our question essentially is, are these independent species? They do correspond to different mitochondrial lineages. So both the mitochondrial sequences and the genomic data pull out the same groups of bees as distinct. So there are now a lot of methods for species delimitation, deciding which populations are distinct species based on genetic data. And I can tell you that these methods are numerous. They're kind of contentious. Some are known to be much more likely to split things into multiple species. Other methods are much more likely to lump things together in one species. So we used several methods in our study. Structure, which basically is looking at admixture between populations, gene flow between populations. Bayes factor delimitation, which tests alternatives to determine likely species based on models of coalescence. So tracing genotypes back to a common ancestor on a time scale based on models of sequence evolution. It takes incredible amounts of computer time. And then DAPC, or discriminant analysis of principal components. I'm going to show you this one primarily because it gives you very nice graphics and shows what's going on quite clearly. So DAPC is a method that combines both principal components and discriminant analysis. So principal component analysis tries to find the set of variables that maximally explains variation among individuals. Discriminant analysis tries to find factors that maximize differences among genetic cluster or among clusters. And so this combined method first finds essentially factors that explain the maximum amount of variation among individuals, like principal components would do, but then takes those factors, which are actually combinations of alleles. What combinations of alleles explains most variation, and then treats it in a discriminant analysis to find out what clusters are best supported, what clustering of individuals maximizes differences among the clusters. So this first illustration is on a large scale, and you can see that we have basically three very distinct groups. Apis mellifera, not a surprise. Kashevnikovi, one of the earliest species, one of the results of the earliest species divergence within the Asian cavity nesting bees. And then this group, which is populations currently or very recently placed in Cerana. So we have Nigricincta, which is now recognized as a distinct species. We have our Philippines population, which is, according to this analysis, it's actually more distinct from the other mainland and Sundaland populations than even Nigricincta is. And then this pileup is the other populations currently placed within Cerana. So India yellow bees, India black bees, central and southern mainland Asia, northeastern mainland Asia, Sundaland. So the next figure focuses in on those currently placed in Cerana, with the exception of the Philippine bees, which we already know are very close to Nigricincta. So we pulled out Nigricincta and its sister group. And so again, we have three very distinct groups: the India yellow bees, the Sundaland bees, and these are all populations in that mainland mitochondrial lineage, northeastern Asia, the south and central mainland Asia, and then the black bees of India. So nested within the mainland group. So what these data indicate to us are that we have good support for the species status of the India yellow bees, the Sundaland, and the mainland group. And I can tell you that the other two methods that we used for species delimitation, the Bayes factor delimitation and structure, agreed with this DAPC analysis. We've got moderate genetic support for this species status of the Philippine bees. So the DAPC analysis indicated that they were equal to Nigricincta or even more deserving of species status on a genetic basis. But the Bayes factor analysis did not find that. The Bayes factor analysis supported grouping Philippine and Nigricincta as one species, which is kind of interesting because Nigricincta, we're always looking for morphological differences. Nigricincta has a very distinctive one in that, unlike all the other Asian cavity nesting bees, Nigricincta lacks a little pore, a little hole in the cap of the drone brood cells. So everybody else has that. Nobody knows exactly what it's for. And the ones in the Philippines, we specifically went back and asked that question. They have the pore just like the others. So there is a physical difference between these two, which would suggest they should be considered separate species. But in any case, they are very closely related and they're not part of the other group. So what does that mean for Indian honeybees? Well, in a nutshell, summing everything up, we see that if you look within the dwarf honeybees, it's been proposed by previous studies that a speciation event that led to Florea and Andreniformis probably took place around the region of the Arakan Mountains, south of the Himalayas, and somewhere between extreme northeastern India and Myanmar. And within Florea, we see another apparent separation of populations somewhere between India and Thailand, so probably in this same region. We don't have samples from this critical region for the dwarf honeybees. And in India, the Florea that's present, at least through southern India, is of the Western type. And if we talk about the giant honeybees, we see again the bees from the south of India are genetically distinctive from Dorsata. Oops, I just changed its range. How about that? Genetically distinct from the giant honeybees on the rest of mainland Asia. But again, we don't know exactly how far the range of this population extends. And this is purely based on genetic data. So far, we don't have any knowledge about morphological distinction between the two populations. When we come to the yellow and black bees, the yellow honeybee has been proposed as a separate species many times by several different authors. And the black bee is known to belong to the mainland Asian population, whereas this yellow population, the plains bees, we have samples from southern India, Sri Lanka, and the Andaman Islands showing this genotype, and both in mitochondria and genomic DNA. So it appears, again, another example of where we propose that India, again, while isolated climatically probably from the rest of Asia to the east, diverged, those cavity nesting bees diverged to give rise to this yellow plains form. And then more recently, a back migration of bees from mainland Asia back into India. So when we look at the black bees of India, they're clearly nested in the group of all the other mainland Asian cavity nesting bees, but the genotypes that we find in India have so far not been found in elsewhere in Asia. So that migration back into India was long enough ago for them to diverge and develop independent mitochondrial sequences. So we presume, well, historically, this implies that India was periodically isolated from Asia to the east and north, certainly by the Himalayas and probably by the Arakan mountain range to the east. Indian Apis species diverged from the East Asian Apis species, or you could say vice versa. They diverged from each other, and possibly this happened multiple times. And so there's just so much work that would be just wonderful to do. Collect and barcode, basically, for a starter. Find out where's the boundary between Eastern and Western Florea. Sampling in extreme northeast of India for the dwarf honeybees. Do they overlap at all with the Western type? If so, it'd be interesting to do comparative studies of their behavior. What's the distribution of the southern Indian honeybee? We have samples of Dorsata bees or giant bees from the south of India, and then up north in Pakistan, Nepal, and Assam. There's a lot of territory between those, and we don't know the ranges of these bees. And as somebody pointed out earlier, these are migratory bees. So this opens up a whole new area of research. How do these migration routes differ? And finally, what are the exact ranges and areas of overlap between India black and India yellow cavity nesting bees? And this is one that is a bit urgent in the sense that people do move Serrana bees around. So some of the original geographic regions may be getting blurred by people transporting bees. So in this case, the sooner the better. We can ask questions for all of them, like what parasites do they host, mating times, pollination preferences. So knowing that there's a lot of diversity and places where you either may have multiple types, multiple species of these bees in India, or at least forms that are different from those in Eastern Asia, opens up an arena for a lot of comparative studies. And so I really have too many collaborators to even fit on one slide, but for this talk, I especially would like to thank my former student, Chet Bada, Nicola Bradbear, Ben Oldroyd, and Gard Otis, whom many of you know, and two other former students, Young Chao Su and Natapat Warren, who helped with discussions, collections. Young Chao and his students now carried out the computational analyses for the genomic study of cavity nesters. And of course, I'd like to thank the bees who gave their lives for this study. So thank you. And that's that.
Thank you very much, Deborah. That was a very interesting talk, and I think there should be a lot of questions. So we are now opening the question and answer session. Let me start. Your talk today, yesterday, paper in Indian journal talking about brown bees, brown Apis bees. It was in Tamil Nadu. I think there's it might be people from Tamil Nadu, because so if you collected them in Bangalore, most people kind of collected in Bangalore and to the Western Ghats, but not much was towards Tamil Nadu. And it would be interesting, I mean, if it's true that they are brown would mean hybrid, right?
Well, so, I thought so too, actually, but I did look at some that brown bees.
Okay.
And so I had some nice yellow ones and some very old ones. So I do have some dating back to the 1990s, is where a lot of my samples from an IUSSI meeting that was held in Bangalore. So, so these bees, I had yellow, I had black, and I had brown. And at least in terms of the mitochondrial genome, all of the brown ones had the yellow mitochondrial genome. So I mean, there's many explanations. You know, one is that, well, yellow bees can come in brown, or at the other extreme, you could say maybe they are hybridizing. If so, it looks like it's a one-way event if we're only finding brown where the mother was, because the mitochondrial DNA is maternally inherited, all of these brown ones have yellow mother. So if it is hybridization, it doesn't seem to be complete. And that's why it would be really fun to do more genomic studies where you would pick up both male and female input. I suspect it may be that there's color variation within the group of the yellow bee species, but until we actually get data, we can't say definitively.
Okay, thank you. So, Lavadi?
Do you hear me?
Yeah.
Yeah. Deborah, that was a wonderful talk. It was an eye-opener because we take too many things for granted with the honeybees, with reference to identification. And I was just wondering, the variants that you have observed with Sarana, can they be addressed now for the time being called subspecies of Sarana?
Well, at the moment, I have a lot of trouble with subspecies within honeybees because there's a long history of, and Michael Engel has a paper on this where he tried to straighten out subspecies terminologies, and there's a long history of people just throwing subspecies names
because
out.
Already many people have used Sarana, Sarana, Sarana indica, and all that. So that's the reason why I'm asking.
And if those are actually fairly, at least in the world of taxonomy, those names were have a good history, you know. But if this I believe if the yellow bee is formally described as a species, it will be indica. Just be Apis indica. But I'm always I always hesitate to say something is such a subspecies because I just don't want to throw another subspecies name out into that crazy world of subspecies names. There are some of them named with so little support, and then the names get picked up and used widely, and sometimes not with much support. So
I think it needs much more field work, I think.
Pardon?
Yes. It needs much more field work, I think, now.
Yeah, field work really to find out what's happening on the ground. What is
true.
Yeah.
Okay, thank you.
Okay, are there any other questions? Question from Professor Butt on YouTube that goes back to Dosata, and he says that for Dosata is in the dusk, right? And therefore it might be that the drone congregation area goes to the nest location clusters, and that this close range mating system might inhibit or kind of like work against kind of mixture of northern and southern Dosata.
Yeah, I don't, again, I have only been in India a few times and haven't had, long field seasons there. So I don't, I can't really claim to know the biology of them well but in India, certainly if they're drone congregation areas, if they were drawing from more close regional populations, that would limit long distance gene flow and push them towards being less mixed. But I don't have any data one way or the other.
Let's try Paul. Paul, you can try to ask your question. Paul? He's not responding.
Oh, yes, I can hear you. Oh, come back.
I'm sorry. I think that the delay is about 20, 30 seconds. It's not going to work.
Say the question. Just. If you say the question, we can answer later.
I'm sorry. I think that the delay is about 20, 30 seconds. Say the question. Oh, I was just really interested in the work you've done on using coalescence I was really interested in the work you were doing on coalescence to look at species as evolutionarily independent lineages. And it would be really nice to try some of the things like Poisson tree process analysis.
Yeah, this is where I would call in my former student, Young Chao Su, who, you know, I'm good at doing the chemistry side, and he's my computational partner. So, and he is desperately interested in trying every new method that comes along. So we may get to that with our next study.
Waiting whether he is
that I think, yeah, I drew the line at getting involved in the massive data analysis and the computational methods. I decided that was for somebody.
Oh, sorry. I think this is taking too long. I will send an email, and it'd be very interesting to have a chat about this further. Thank you.
Yeah.
Thanks, Paul. Comment, Dr. Saravanan, on the brown bee. In terms of the Serrana black type. Oh, wait a minute. Okay, I read it. Whatever. In Tamil Nadu, the Apis Serrana black type are confined to high altitude, above 1,200 meters. The yellow ones are present only in plains of the Peninsula India. How does this kind of variation occur? Your opinion, please.
Well, I mean, if they truly are different species, which I believe they are, it's not unusual for species to have different optimal habitats. And so, you know, part of it could simply be that these are optimally adapted to either warmer, drier, or cooler, higher temperature conditions and ecosystems. And there could be a historical component to it too, simply that this may be the yellow bee may be a more, this is where I really want to get some genomic data rather than just mitochondrial, and better sampling through that area. Indonesia is a big black hole at the moment for getting specimens. But it may be that the Indian bees, yellow Indian bees, are more allied with Sundaland populations than with the bees to the north, with the mainland population. So part of their distribution may just be historical as well. But heading up north into India, I don't know how far north the yellow population extends or where it extends to. And that would be a good project for systematic sampling, just to see at least where they occur today.
So one question for me in addition. So you think that the border for the yellow bee, so there's a border of the yellow bee to the north, and how about the black bee, kind of like to the south?
Well, likewise, I don't. I know the black bee comes all the way down. Certainly, it's in Bangalore, it's in Karnataka, and the Western Ghats. So it seems to have come in, and it's not limited to the north by any means.
Okay, let me kind of like, I have to read the questions. So Professor Bhatt asked a question, but you have already answered it, but I will read it. Black and intermediate color. Probably yellow interbreeding. What is your opinion? You already answered the question? Hello, Deborah. Thanks for a nice presentation. That's by Padma Kumari. Can you please suggest which species is more sensitive to insecticides, as safety is one of the criteria for green pesticides?
Yeah, no, I can't make any suggestions because I really don't have field data on the behavior or physiology of the bees. But it's there's so many things that would be interesting to look like their thermal tolerance limits, for example, as climate warming becomes more and more serious, temperature regimes may start to affect the distributions of many species, including honeybees, and even knowing their tolerance limits for high temperatures, do they differ? I mean, given that the hill bee is typically at, according to what we know, somewhat higher altitudes, the plain bee at lower, although that's not 100% true. You can find them side by side sometimes. Does the black bee have lower tolerance to extreme temperatures, or does the yellow bee tolerate higher temperatures? Would be interesting to know.
Thino just raised his hand, and because he is from Kerala, maybe he wanted to say something about the south.
Can I ask a question? Am I audible?
Yes.
Yeah, Deborah, this Sri Lankan one is it fully yellow, or there are black ones still there that you have not sampled? Because when you were presenting this, you were telling the black one is in the high altitude and the yellow is in the plains, and Sri Lanka is a fully yellow Apis cerana. So what is yeah.
Yeah, the number of specimens I have from Sri Lanka is very limited, but all of them have been of the yellow type. So I couldn't say definitively that there's no black ones there, but of the small samples that I have, they have all been of the yellow type. So I know the yellow is there. At this point, with the data I have, I couldn't say that there's no other cavity nesting bee there, that there's no black bee. But someone...
Deborah, does that tell that in other parts you may have, I mean, not in the mainland Asia, but in other northeast, and not northeast, southeast, they may have this black or brown, all this, but they are not sampled well or not present in your sample.
Yeah, I mean, to be honest, my samples, as you saw on the maps, are limited. I have the Bangalore, Karnataka, some from Kerala and Western Ghats, but then a big jump up to Nepal. And, I simply don't have any material between those places. It would be it's the kind of project that needs to be done on the ground, needs,
Yeah, that's a future project, Deborah. Thank you so much. Yeah.
Okay, Deborah, still some few questions from YouTube. Dr. Avinash Chauhan. Thank you for the nice presentation, Dr. Deborah. Any linkages that separate Apis florea of northern eastern India and mainland India? It means kind of like whether there's a geographical barrier.
Well, again, yeah. What the one that people have pointed to first in the context of the two species Florea and Andreniformis, they point to the north-south running mountain chain, Arakan, that would sort of cut, put in the rest of India under a little cap from the Himalayas and then coming around to the east. So if it became either too cold or dry for open nesting honeybees, those mountains could be especially the lower Arakans, could become formidable barriers. At least that's the hypothesis. And, with the eastern and western Florea, given the samples we have, it looks like that same region in between Assam and Myanmar, somewhere in there, is the barrier, and that's where the Arakan Mountains are. So I logically guess that is possibly has been a barrier more than once to flow of organisms back and forth.
Okay.
Mhm.
Okay, so this is now a terminology question from Sarah Vanan. Can the species variation be called ecotypes, or both black and yellow are distinct species?
I think clearly the black and yellow are, this is one where I feel more comfortable saying, yeah, the yellow one's a species. It's not only me, many people have looked at different aspects of the yellow honeybee compared to the black hillbee and found differences in mating flight times, in, we have a variety of genetic differences. Early on, we did allozyme differences. And I would say the data is pretty convincing, but what we lack is when you're doing a species description, they like something other than color, because color can be very variable within an insect species. And so, some of the studies like Axel, you and your group were doing on Laboriosa and Dorsata, just a concentrated, let's look at the morphology in detail. Something like that would be what you would want to do for the yellow and black bees, not just the morphometric differences, which again, morphometrics have shown differences between the two populations as well, but something qualitative, like a genitalic difference, or as you were saying with the Laboriosa versus the other giant bees, differences in the ocelli, some physical differences would make it easier to do the formal species description, but they may have different ecological preferences, that's true, but it's not like if they were ecotypes of one species, you would expect them to be each other's closest relatives, and they're not. The yellow ones are actually quite distant from the black ones. The black ones are well nested in the big Asian mainland population. So they have different ecological preferences, but they're not ecotypes of one species. Yeah.
Because I'm coming from olfaction, wondering whether studies on the queen mandibular gland would be also interesting. Many people think always of mating flight times, but speciation is often also done by changes in the pheromone communication.
Mm-hmm, mm-hmm. That's what I get fascinated by the biogeography and the potential for speciation events. But really, one of the advantages of having that insight is that it opens up all sorts of new comparative studies. There's so many questions now and actually you can make a lot of mistakes, like a lot of early research on Varroa mites before people realized the diversity of Varroa mites, not only that there were multiple species on that had invaded Apis mellifera, but also, I mean, that there were multiple species of Varroa and only one tiny population had invaded mellifera. A lot of that early research on how does Serrana handle the parasite versus mellifera, that research is not showing at all what the authors thought, because they didn't realize they would come. There is a lovely set of studies by Peter Rosenkraus. When he came to India early on, he was wanted to see how the workers of Serrana and mellifera handled introduced mites. So, of course, on the natural host, it's hard to find Varroa. The natural host keeps them rare. So he got all the mites from mellifera, which was, now we know it's not even the same species as what's in India. So he did a nice study, but it was a study on how does the Indian Serrana handle a foreign mite, not how does it control its own mite. So
yeah, okay, I mean, this is good, not because most research in India focuses on Apis mellifera and Apis serrana, right? Mm-hmm. And need to expand that. Mm-hmm. I have to go back to the question. Let me see. So there's one question on YouTube, also by Professor Butt, and I think it's a question about which of the honeybees came first, the open nesting or the cavity nesting? You know, there's always this, it's going back to Nicholas Koeniger.
Yeah. Nicholas and I have a book chapter we wrote together, and with Gudrun Koeniger, and that was so hard for me because, I mean, the argument that, I mean, Nico Koeniger's argument is that the ancestral form of honeybees should be the cavity nesting ones, because, well, most bees are ground nesting. So that, and of their relatives, you know, he says, well, look at bumblebees. Bumblebees are ground nesting. But basically, if you look at, well, all of the genetic data puts it the other way, that the open nesting ones are at the base of the tree, the open nesting dwarf bees, and then a split to the giant and cavity nesters. And so cavity nesting is derived in these guys, in mellifera and the Asian cavity nesters. So we had an interesting time writing a chapter together on evolution of the bees, but it's a perfectly logical starting hypothesis. It's just that also looking at the dance language, you know, in the open nesting bees, you see a more direct use of the sun cues, and then conversion of the visual view of the sky to the symbolic dance on a vertical comb. And that certainly wouldn't make sense if that's the original population or original way of life of the bees. So, I mean, most of the data don't support the hypothesis that cavity nesting was the original form for Apis.
One question. Vidua?
Yeah, hi. Am I audible?
Audible, Vidua.
Yeah.
Okay, cool. So just a quick context setting and then two questions. So I'm So I'm actually in Axel's lab doing my PhD, or at least the initial part of it, on black and yellow Sarana. And now some Sarana colonies we have on campus in NCBS. We sent pictures of them to an alumnus of the lab for IDing, and she seemed to say that they're a hybrid of black and yellow, similar to the bees used by the adjacent agricultural university. And we also, I guess we discussed earlier in this talk that the brown bees might be a hybrid of black and yellow. So if these two things are hybrids, these two aforementioned things, then like how does that affect the whole question of whether the black and yellow, at least in South India, are distinct species? And my next question is, if the black bees in South India are a result of recolonization by mainland Sarana, I mean, then what like geographical factors or changes actually enabled them to do that recolonizing? So that's my questions.
Okay, well, I'll start with the last question first. This is where I am hoping, actually, we want to do another study of the cavity nesting bees with, we hope we can get even better geographic sampling, but also get more precise dating, because all of our dates on our tree have fairly broad error bands. But if we can feel confident, more confident about the proposed times of speciation events, it makes it easier to pin down a particular geological event or climatic event that might be associated with different events. So like invasion of black bees from the east coming into India, I would expect that it was, well, actually there's two hypotheses. It could be a period of warmer weather that enables them to easily get over barriers like mountain ranges, or more intriguingly, it could be very cold episodes of glaciation where if sea levels are lower, some of that land along the coast that is now submerged, an arc between Myanmar to India, those low sunken lands could have been dry land and lowland that might have been passable by bees. So that's where, you can make a lot of hypotheses, but without better data on the timing of events, it's hard to pin down exactly which alternative might be the correct one. But as far as the brown bees, again, I can imagine two remember my data, let's put it this way. Both the mitochondrial and the genomic data from the black and yellow bees shows them to be quite different, both not only from the mitochondrial data, but also the genetic material inherited from both parents. And it also shows that the black bees are, clearly that mitochondrial ancestry, and the nuclear genome is grouping with all the rest of Eastern Asia. So I'm fairly confident that they are separate species. You can have hybridization can take place between species. And I think it's interesting that all the brown bees that I have looked at, their mitochondrial DNA was yellow. In fact, I did not include any of those in the genomic studies. So next time we do it with better techniques, more modern, more modern techniques, we will include some brown bees to try to get at that question. Are they hybrids between black and yellow, or are they simply a brown? Now there you could say ecotype, a brown ecotype within the yellow species, the Apis species. At the moment, I'm leaning, well, at the moment, I guess I should say the data aren't there to make a 100% clear, decision, but I'm leaning towards it's all, maybe color variants within the within the yellow species. But.
Okay, cool. Thank you.
Deborah, we are almost 2 hours, but let me, I have one question. That's the last one. So as you know, we collected Dosata from the Andaman Island, right?
Mm-hmm.
So the Dosata, if you are smaller than on the mainland. So when we kind of included all the size-dependent characters, we got them separated. If we remove size-related characters, also going morphological with the mainland. So the size difference is more a plasticity phenomenon, right?
Yeah.
Would you have any idea? The cells of the combs might be smaller, or?
Well, I guess I'm not as well versed in how island populations of insects respond in terms of body size. I've read the papers talking about, island populations leading to, gigantism of normally small creatures and dwarfing on normally large. So you have the, islands with the pygmy hippos and pygmy elephants and giant examples of grasshoppers in New Zealand taking the niches of other organisms. So I don't know what about an island environment might lead to smaller that doesn't bother me. The ones that we looked at from the Andamans grouped, now that I have to remember that, grouped with the giant bees we found from southern India. I think that's true. Yes.
That's an interesting question in itself. So let's stop, because kind of like people are also kind of reducing, and it's 2 hours. It was a very nice talk. One of the comments was on YouTube was, I think we should hope that in the future you will come and visit, or we can invite you. That it's obviously clear that there are overlapping interests, and you are the person to go to about the phylogeny. So
and India is really, it's going to be a really interesting place. It is an interesting place to work on honeybees because of the diversity and the possibility for these interesting comparative studies. So it's
okay. Good. I thank you once again.
Thank you.
Thank you. We don't hear from the others. And then my last task today is to announce the next week's speaker, and that is Professor Christina Grosinger from the Center for Pollinator Research at Penn State University. And it also starts at 6:30, if I'm correct. And she will talk about supporting pollinator biodiversity through transdisciplinary programs. So a little bit of different topic.
And that should be a good talk, yeah.
I'm looking forward to that. Okay, thank you very much. And then I have to say goodbye.
Okay, bye-bye.
For you, Deborah.
Okay, thank you. How do I leave? There we go.